If there is no anticoagulant inside this tube, why would a laboratory actually want the blood to clot? The red top tube is a standard vacuum collection device engineered for a single specific purpose: to obtain serum. Biologically, serum is the liquid portion of blood that remains after the whole blood has been allowed to fully coagulate. When you look at a spun tube of serum next to a tube of plasma, they appear identical. Both are clear, yellowish fluids. The distinction lies entirely in their biochemical makeup, specifically the presence or absence of clotting proteins. Plasma is derived from blood that is chemically prevented from clotting. Because the coagulation cascade is blocked by an anticoagulant, all the original blood proteins, including fibrinogen, remain intact and suspended in the fluid. Serum works in reverse. During coagulation, the developing blood clot acts like a biological sponge. It physically consumes fibrinogen and numerous other clotting factors, stripping them out of the surrounding fluid and leaving a much cleaner matrix behind. To achieve this, the laboratory intentionally allows the blood sample to destroy its own cellular suspension. This controlled clotting process removes protein interference before the final analysis even begins.
Want to dive deeper into the amazing world of microbes? Then join our community and become a member today. And don't forget to check out our new microbiology study app.
The primary target of this removal process is fibrinogen. During normal coagulation, fibrinogen converts into solid fibrin threads to form the structural scaffolding of a blood clot. Inside a red top tube, this chemical conversion is allowed to run to absolute completion. The liquid is essentially cleansed of these large reactive proteins. Therefore, serum is defined by what it lacks. It is blood fluid minus fibrinogen and the coagulation factors consumed during the clotting cascade. This biochemical divide dictates how laboratories process samples. Choosing serum over plasma is a strict requirement to maintain diagnostic accuracy in specific clinical tests. To facilitate this process, red top tubes are manufactured in two distinct variations: traditional glass and modern plastic. A plain glass tube contains 0 chemical additives. The physical surface of the borosilicate glass provides a negatively charged boundary that directly triggers the blood's intrinsic coagulation pathway. Modern plastic tubes, made of polyethylene terephthalate, present a problem. The plastic is chemically inert. Without help, blood sitting in a plastic tube takes far too long to clot. To solve this, manufacturers spray coat the interior walls with a clot activator made of microscopic silica particles. These tiny glass beads provide the necessary surface area to trigger rapid coagulation. Because this activator is a physical additive, the blood must interact with it. A plastic red top tube must be gently inverted 5 to 10 times immediately after collection to ensure the silica distributes evenly throughout the sample. The plain red top is a precision-engineered environment designed to force a biological reaction, whether it relies on the physical properties of glass or dispersed microscopic particles. This forced reaction operates on a strict timeline. A plain glass tube requires a full 60 minutes of upright standing at room temperature to complete the cascade, while a silica-activated plastic tube cuts that time to 30 minutes. During this window, a process called clot retraction occurs. Platelets trapped inside the newly formed fibrin mesh contract, physically squeezing the liquid serum out of the solidifying cellular mass. Once the biological clock finishes and the clot is secure, the specimen must be centrifuged to isolate the serum. The high relative centrifugal force pulls the dense cell-packed fibrin clot to the bottom of the tube. The cell-free, fibrinogen-depleted serum is left resting cleanly at the top. This highlights the main mechanical drawback of serum. Plasma can be centrifuged immediately upon collection without any waiting period, making it the preferred matrix for rapid response emergency testing. Serum collection demands patience. Skipping or short-circuiting the clotting wait time compromises the physical and chemical integrity of the entire specimen. The reliance on a pure serum matrix traces back to the early days of clinical chemistry, where analyzing the distinct protein bands in human blood became a critical diagnostic tool. One of these tools is serum protein electrophoresis, or SPEP. This assay separates blood proteins by size and electrical charge, and it strictly forbids the use of plasma. If a lab runs plasma through an SPEP, the intact fibrinogen migrates to form a sharp, narrow band on the readout. This artifact mimics a monoclonal immunoglobulin band, causing a false positive for severe disorders like multiple myeloma. Plain red tops are also required for therapeutic drug monitoring. Assays for certain medications, such as the anticonvulsant gabapentin, mandate a plain tube rather than a gold-top serum separator tube. The SST utilizes a polymer gel to separate the cells from the serum. However, this gel can absorb specific drug molecules out of the liquid over time, resulting in falsely low concentration readings for the patient. The lack of a barrier gel or anticoagulant makes the red top tube a chemically neutral environment, preserving the exact concentrations of highly sensitive analytes. When comparing red tops to other matrices, the trade-offs become clear. A green top heparin tube yields plasma quickly, but heparin inhibits the polymerase chain reactions required for DNA analysis and interferes with certain chemical stains. A lavender top EDTA tube preserves cellular morphology perfectly for hematology, but EDTA works by permanently binding calcium. Running an EDTA sample on a chemistry panel will result in an artificially near-zero calcium reading. Volumetrically, serum yields about 15 to 20% less fluid from the exact same whole blood draw compared to plasma, because a significant amount of liquid is permanently trapped inside the clot. Despite the lower volume, serum remains the gold standard for measuring clinical enzymes. Anticoagulants bind the specific divalent cations that many enzymes require to function, which suppresses their apparent activity on the analyzer. Selecting a tube is a strategic calculation. The phlebotomist must match the physical matrix of the sample to the chemical requirements of the target analyte. When red top specimens fail, it is usually due to handling errors. Vigorously shaking a plastic tube instead of gently inverting it shears the red blood cells apart. This hemolysis turns the serum pink and releases intracellular potassium, completely invalidating electrolyte panels. The second major failure is latent fibrin, occurring when a technician places the tube in the centrifuge before the mandatory 30-minute clotting window elapses. If spun prematurely, the coagulation cascade continues operating in the separated serum. Stringy, invisible fibrin webs form inside the liquid, causing mechanical clogs in the aspiration probes of automated analyzers, leading to immediate specimen rejection. These pre-analytical errors stem from a failure to respect the biological clock of the coagulation cascade. For laboratory professionals, mastering the red top comes down to 10 essential facts. 1. Red tops yield serum. 2. Plastic tubes use silica as a clot activator. 3. Plastic tubes require 5 to 10 inversions. 4. Clotting requires a 30 to 60-minute wait. 5. Serum lacks fibrinogen. 6. Plasma retains fibrinogen. 7. Intact fibrinogen in plasma causes false M spikes on electrophoresis. 8. Plain red tops are used for therapeutic drug monitoring to avoid gel absorption. 9. Premature spinning causes latent fibrin strands. 10. Shaking causes hemolysis. These rules form the absolute foundation of pre-analytical quality control. The red top tube utilizes the body's natural hemostatic processes to separate its components. By intentionally triggering coagulation, the laboratory consumes reactive proteins, isolating a pure, unadulterated serum matrix. If you found this lesson useful, subscribe and follow for more MLT topics, laboratory techniques, hematology, microbiology, clinical chemistry, and exam-focused content. In the clinical lab, the quality of the answer depends entirely on the quality of the sample.